So let's get going. Again, thank you for joining this webinar entitled Gene Therapy is Robust Scientific Inquiry, the Missing Factor. We have a large number of registrants from every continent on the globe. My name is David Page, and I will be moderating the session. Note that we are in webinar mode, so your voice can't be heard and your faces won't show on the screen. So you can mute yourself and leave the camera off. The session will last 1 hour, and the final 30 minutes or so will be dedicated to your questions. Before I introduce the agenda and our panelists, please note that you can ask your questions by clicking on the Q&A button at the bottom of the page and typing in your question. You can also show your interest in other people's questions by voting for them and pushing them to the top of the list. We may not be able to get to all the questions. There's also a chat box. If you have any kind of technical questions, difficulties in connecting, if you want to make comments on what you're hearing, say hello to your friends, please use the chat box. Note that the interest in holding this webinar arose after the publication of this commentary in the journal Hemophilia in July of this year. So our goal today is to have a very accessible discussion of the key issues that were raised in that commentary, and we're going to have a minimum of detailed clinical trial data, charts, and graphs. So we're very fortunate in having almost all of the authors of that commentary on our panel today. The only person not with us is Declan Noon, chair of the European Hemophilia Consortium, and we miss you, Declan. So our panelists are Glenn Pierce, Vice President Medical of the World Federation of Hemophilia. Glenn has extensive experience in gene therapy research. Radoslaw Kaczmarek from the Polish Hemophilia Society is also a researcher in coagulation therapies and currently chairs the World Federation of Hemophilia Coagulation Products, Safety, Supply, and Accessibility Committee. Brian O'Mahony was president of the World Federation from 1994 to 2004 and is past president of the EHC and is currently the chief executive officer of the Irish Hemophilia Society. And Mark Skinner, WFH president from 2004 to 2012, is involved in too many projects to name, both in the United States and internationally. So collectively, I calculate that the group has approximately 300 years of experience living with hemophilia A or B. That works out to a mean of 50 years per person. So ignoring variability, and by my advanced mathematics, I will claim to be 50 years old for the rest of the session. To set the stage and to make sure we all have a basic understanding of gene therapy using the adeno-associated virus vector, I'll ask Glenn Pierce to get things going. Over to you, Glenn.
All right, thanks, David. And it's a pleasure to be here with my colleagues today. I'd like to do some level setting and get us all started in the right direction with regard to gene therapy. Gene therapy is a means to the end. Our goal is to see hemophilia cured, pure and simple. And how do we do that? Well, there have been many attempts at it, and the most current one over the past 25 years has been the use of AAV, adeno-associated virus, which we have been able to vectorize, to remove its genes and put in the factor VIII or factor IX genes. Over the years, scientists have figured out that they can just infuse it intravenously. If we move on and build the slide, please. Once it's infused intravenously, most of the AAV, regardless of the serotype, goes to the liver. And in the liver, there's a process called transduction. And all that means is that that's the way that the virus enters the cell. The cell in question is the hepatocyte, which are the factories in the liver that are responsible for so much of the work that occurs within the body and the synthesis of all of the proteins, many of the proteins in the body. What happens once the virus is in the cell is pretty much of a black box, but what we know is that a little bit of it integrates into the chromosomes and remains there forever. Most of it, though, is destroyed as it goes into the cell along the way. Another small amount forms what are called episomes, and those episomes are circular pieces of DNA that are stable and remain within that cell for long periods of time, many years. And in that stable form, they can take advantage of all of the cell machinery and make protein. And that protein can then get secreted, as you see on the right-hand side of the slide, as either factor 8 or factor 9. Now, of course, it's not that simple. Lots and lots of work have gone in. It's been an iterative process, starting from very bad data long ago in animals to better and better and better data along the way, to the point now that most individuals who are getting AAV gene therapy who have hemophilia A or B are getting some type of a response, often a therapeutic response of some nature. And so that's a very encouraging step going forward. Next slide. There, however, have been many questions that have come up with regard to gene therapy. And as we make more progress, we answer a few questions, but for every question that's answered, there are more questions that come up. And so there still is quite a bit about this entire process that we don't know. This is a framework that Alfonso Iorio and I put together a couple of years ago now, and it's been a useful one, especially when we try to understand what we know and what we don't know about the unknown questions. And so if I can just take a moment and stay on the right-hand side of this 4x4, if we look at the upper right quadrant, this is the known unknowns, and it's what we know we don't know. So we know enough to ask the question, but we don't know enough to know what the answer is. And that's where, as we make continued progress, more and more questions are coming up. The bottom right box is a disturbing box because for us and for anybody else that's investigating new frontiers of science, this is the unknown unknowns. And it's what we know, what we don't know, we don't know. These are the questions we can't even imagine asking, but they will come along as they do for every scientific program and surprise us along the way. And so they're important for us to try to anticipate and keep watch on. So that's the framework in which we came up with this, this idea for this article and now this webinar. And I will turn it back over to David to get into the details of these kinds of questions. David?
Thanks, Glenn. Our paper identified 7 abilities or abilities and knowledge gaps. So eligibility, predictability, tolerability, durability, ability to redose, visibility, and affordability. So our next speakers are going to tackle these topics one by one. So our first panelist is Brian O'Mahony, who will share his thoughts on the first of these concepts, eligibility, or who can receive gene therapy. Over to you, Brian.
Thank you, David. So a significant proportion of people with severe hemophilia are not eligible for gene therapy during the clinical trials. These include children. So the gene therapy currently is only for those over the age of 18. Having said that, there has been a lot of discussion about reducing the eligibility age from 18 to 16, down to 15, and perhaps even as low as 12. The big barrier here is the extent of maturity of the liver. But by age of 12, the liver has reached 2/3 of its adult size. So it's possible to look at gene therapy between the age of 12 and 18, perhaps in coming years. But of course, at that age, you also look at ethical issues. You look at issues of consent and assent. You look at perhaps needing higher doses for children. You're looking perhaps in the longer term at look at integrating vectors. So at the moment, children are excluded. At the moment, apart from the Unicure AMT-060 and AMT-061 trials, those with pre-existing antibodies to the AAV vector serotype being used are also excluded. And that can be anything from 30% with AAV5 to 52% with AAV2. So between 30 and 50% of people can be excluded on that basis. Those with inhibitors, either a history of inhibitors or a current inhibitor, are also excluded. And those with active hepatitis C, hepatitis B, or with some trials, HIV, which is not controlled, are excluded. So that excludes a significant proportion of people from the trials. If we look at the severe hemophilia population, and I looked at all those exclusion criteria and did some figures on the European population from 18 countries based on all the parameters reported to WHO Annual Global Survey. And my estimation is that in Europe, for those 18 countries, only 44% of those with severe factor VIII deficiency are eligible, and 48% of those with severe factor IX deficiency. And when you then go on to a licensed gene therapy, perhaps those with a history of inhibitors may not be excluded, but there'll be other criteria coming into play, such as clinical history, preferences, the individual's treatment goals, their support network, their expectations from the treatment, their adherence history, and perhaps lifestyle factors such as the extent to which they use alcohol. Back to you, David.
Thanks, Brian. Our next speaker is Radek Kaczmarek, and he's going to tackle the concept of predictability, which also encompasses variability and reliability. In other words, can we predict who will have a response? How and why does it vary from person to person? Radek?
Thank you, David. It's a pleasure to be with you all and discuss these again. The short answer to that question is we cannot predict who will have a response to gene therapy and how good that response will be, should there be one. In clinical trials for hemophilia A and B, we see huge variability in factor levels, and some patients fail to achieve any factor expression whatsoever. Those who do achieve long-term expression can end up anywhere from a moderate and mild hemophilia factor levels to normal range to well into supraphysiological levels. And we have no way of knowing who will hit the jackpot and who will end up disappointed. And this uncertainty is troubling because with the current technology, everyone will have just one shot at the therapy, and so redosing will not be possible. It is also troubling for other reasons. The coagulation system tolerates large variability in coagulation factor levels in general and factor VIII and IX levels in particular, which you can tell by looking at the wide normal ranges for both, spanning from 50 to 150%. But that tolerance has its limits, and in extreme cases, some individuals may achieve super high expression, even as high as over 500%, putting them at risk of thrombosis, which actually happened for real. We don't know why there is such a large variability in expression levels. There are likely several mechanisms at play related to immune response to the therapy, the target cell for factor expression, and a number of individual genetic differences in the way the target cells permit the vector entry, unpack the transgene, process it, make the transgene product, namely the clotting factor, and move it out of the cell to the circulation. On all of these, we need more research, even if some of those we will not ever be able to control. Thank you.
One question, Radek. Are we sure that the dose each person is getting is actually the dose that is desired, or is there some uncertainty around exactly how much is given in an infusion?
Well, across the board in trials, we see a huge variations in doses used of different vectors. There's a 200-fold difference in doses used across the board looking at individual trials. And people, even with the same vector, people getting the same dose have no way of knowing what factor level they will end up with. So the variability is regardless of vector dose. It's not only vector dose related. Thanks,
Radek. So far we've been talking about how well gene therapy works and for whom. I'll ask Dr. Glenn Pierce to come back and discuss the crucial question of tolerability. How safe is gene therapy?
Thanks, David. So tolerability or safety is, of course, front and center on everyone's mind and is the most important thing to study in a early phase clinical trial, a phase 1-2 kind of clinical trial. We should mention that safety in hemophilia is very good to date with regard to AAV. And overall in the AAV field for the use in many different kinds of diseases, overall the safety record is very, very good, with a few notable exceptions though. And maybe it's best to divide it up in terms of how far out a person is after receiving gene therapy. So after one receives AAV gene therapy, in the immediate 24 to 48 hours, there is a small but definite risk of having an immune reaction to the gene therapy that can be in the form of like an acute influenza syndrome, where there's fatigue, malaise, temperature, maybe a little bit of a allergic reaction. And that probably stems from the fact that trillions to quadrillions of vector genomes are being given to the patient. So more genomes are being given to the patient than we have cells in our body. That's the order of magnitude of the amount of virus that we get. And this is a different way that we're getting this virus. Viral infections normally start off small and gradually build up. This one is a virus that's going in all at once. And so in a few patients, that can cause a reaction. Those reactions have generally been well controlled. There's probably been 1 or 2 per clinical trial that have been seen. So they're rare, but they're not non-existent. Once we're past that period, over weeks to months, the second area of safety that is important to identify and understand is the liver enzyme elevations. Liver enzyme elevations, which are the kinds of things that occur in hepatitis C that many of us are very familiar with over the years, indicate that hepatocytes are dying. And so they can be caused by a variety of different reasons. In this case, they're being caused by the intervention, the AAV gene therapy. What's causing that? That's not at all clear. We know that at least there is a contribution from the adaptive immune response, the immune response we have that actually protects us from viruses. Does the same thing when we are getting the AAV infused. And so that adaptive immune response is designed to destroy virally infected cells. And it's the relationship we've had for 500 million years as vertebrates with viruses that have arisen. Their goal is to infect us. Our goal is to prevent them from doing that. And so that immune response occurs, and that can result in elevations in these liver enzymes. Those generally are not high, although in a few trials they have gone 10 to 20-fold above the upper limit of normal. And those trials have generally been closed down because of that. Most of the time they're 1.5, 2 times the upper limit of normal, not a concern clinically at all, but they do represent damage. And so that's a contribution that we don't fully understand. And then in addition, primarily in the factor VIII trials, there seems to be something else going on, perhaps beyond the cytotoxic T cell response that we see as a part of our immune response. And what that other thing is that's going on is a black box. We don't know what that is. So that's the intermediate question of safety that needs to be further addressed. The third component of that is long-term. So what's the risk long-term? I mentioned at the beginning that we do get a small amount of integration, and it is a small amount relative to the amount of virus that is being administered to a patient, but it's not a small amount in absolute terms. It's millions of integrations that go into the chromosomes of the liver in patients who receive gene therapy. Those have had no negative effects at this point in humans, either for hemophilia or any other trial. And so that's a very good sign. But we also need to understand over the long term, years to decades past gene therapy, will that continue to be the case? Will it continue to be safe? And we'll discuss a little bit later the need to have a long-term registry to be able to follow patients. But so far, that doesn't appear to be a problem. Back to you, David.
Well, and Glenn, we all heard back in August that the FDA in the US, the EMA, held back approval of one of the leading gene therapy developments in hemophilia A. And one of the issues appears to be the unknowns around durability, or how long factor expression will last. Could you elaborate on that?
Yes. So with regard to durability, we have to separate factor 9 from factor 8. And as the data emerge, we have several clinical trials, independent clinical trials from a number of different groups for hemophilia B, for factor 9. And over the years, it looks as if that's durable. The level that you get in your first year seems to be the level you'll have at year 2, year 4, even year 8. And I think that's very encouraging. That doesn't deal with the variability that Radek talked about. There still is wide variability in hemophilia B that needs to be addressed because we don't know what patients will get. Will they get a very low response? Will they get a high response? But in terms of durability, I think we can probably take that off the table for hemophilia B. For hemophilia A, however, it's a very different matter. And the durability question in hemophilia A appears to be more of a generalized phenomenon that is present independently in most or all of the clinical trials that we see for hemophilia A. And so that is a distinction, and it's one that's not understood at all. What's the loss of factor? What is that loss of durability? What causes it? Completely unknown at this point in time. And that's a fundamental problem because if it's unknown, then we can't figure out how to intervene and how to fix that problem. And that's the fundamental reason why we need to understand what is causing the loss of durability so that then steps can be taken to try to address that and repair it. We're not interested in gene therapy that doesn't last very long. That doesn't make any sense. We have great therapies, at least in the developed world, to manage our hemophilia. What we're looking for is a cure, and a cure implies some level of durability. Will it be lifelong? Does it need to last 20 years? I don't have a number. None of us will have the same number, but we know that it does need to last for an extended period of time in order to be considered an adequate treatment beyond the treatments that we have today. And that's the fundamental unknown question that there is no answer for at this point.
Thanks, Glenn. So if it turns out to be true that durability is lacking and expression falls off to a level that's no longer protective, can't a person just get a second dose? Just to discuss this, Brian O'Mahony.
Thank you, David. Well, I suppose if redosing was possible in all cases, then durability wouldn't be the big issue that it is. And in terms of can you be redosed with an AAV gene therapy, currently the answer is no. You really have one opportunity. And I think that there have been a number of theoretical possibilities put forward as to how you might be able to redose with an AAV gene therapy. Companies have been looking at using plasmapheresis to create a window whereby you can lower the titer low enough to allow for redosing, because it must be borne in mind, as soon as an individual is dosed with AAV gene therapy, they develop a very high titer antibody to that AAV serotype. So plasmapheresis to create a window to allow redosing is a possibility. The use of immunosuppression to prevent the high titer antibodies forming, or the use of immunoadsorption to eliminate pre-existing antibodies are all theoretical possibilities, but they haven't been used in any clinical studies to date. So I think at the moment the answer is no, you can't redose. Also, even if scientifically one of these strategies were to work and you could redose, it may be financially unviable to redose because depending on reimbursement systems, countries may decide that you get one opportunity for gene therapy, and if it doesn't work, or if you lose expression and you don't get a second opportunity, it may be too expensive.
Okay, so gene therapy is therefore a single infusion and there's no going back. So visibility or transparency is critical. So what information should be available to people thinking about receiving gene therapy? And to discuss these issues, Mark Skinner.
Hello all, it's great to be with you and colleagues today as well. So Brian's previous comments really are the really the crux of why this is so fundamentally important in gene therapy. The irreversible nature of gene therapy requires the greatest transparency so that patients in combination with their clinicians can actually evaluate between therapies, both those that are existing on the market and those that might be on the horizon, to make a decision: when do I want to have my one opportunity to pursue gene therapy? Am I better off with another therapy? And even between gene therapies, if there's multiple choices, that there is complete transparency in the data so patients can evaluate which might be the better decision. To do that, we really are needing and require consistent real-time communication of clinical trials, and when it becomes available in the market, real-world data, particularly around core outcomes, data free from commercial bias and at a level of detail that allows comparison and interpretation. In 2018, for the first time, the hemophilia community developed a core outcome set for gene therapies. That was a project that was developed and named Core Heme, and it had extensive involvement of patients, clinicians, researchers, drug developers, regulators, and payers. And this was really highlighted by the importance of the patient involvement so that the efficacy outcomes are both meaningful to us as patients as well as relevant to clinical care. And those outcomes in the core set included things like frequency of bleeding and factor activity level, duration that we've talked about, but also other qualitative measures like chronic pain, mental health, and utilization of healthcare resources. But importantly and relevant for today's conversation, that core outcome set also included safety events and a list of safety events and data that should be widely made available and consistently reported across trials, both because of good clinical practice and regulatory guidance, but also because they are important to us in the decision-making process given the nature of gene therapy. So these included many of the things that we've talked about: liver toxicity, the immune response to the transgene and the capsid, thrombosis risks, the development of other potential disorders, vector integration into the host genome, duration of factor neutralizing responses, and in the event it would occur, anything around a cause of death. These efficacy and safety outcomes that we've talked about, that were identified as core outcomes, are as relevant today as they are when they were adopted, and we need the manufacturers to refresh their recollection and consistently and timely report these so that they are available, and where they aren't available, that we have robust inquiry to try to begin to answer those questions. We will talk about it just in a moment, but the World Gene Therapy Registry that the WFH is developing really is an important piece and tool for this. It will allow us in the real world phase, not in the clinical trial phase, to collect efficacy outcomes as well as the patient important outcomes and safety outcomes so that we can compare them over time. And that too will require the WFH to have a platform so that there's real-time sharing of data. I think Brian in a moment is going to speak a little bit more how all of this and the transparency relates to the informed consent process and the shared decision making.
So don't go anywhere, Mark. No matter how well gene therapy works, no matter how long it lasts and how safe it is, it doesn't matter if nobody has access to it. So can you say a few words about affordability?
Sure. And so visibility and transparency aren't just important to us as patients. It really matters in the affordability area as well. Payers, health systems, they need to have confidence in the therapies they're being asked to pay for. They're going to be asked to make a significant investment. So the visibility and transparency of data is fundamentally important as we move into technology assessments. Patients want to know if it is the safest and most effective drug that's ever invented. If nobody's willing to pay for it, it's of no benefit to us as a patient. Encouragingly, the first technology assessment for gene therapy occurred here in the United States. And despite the pullback, the complete response letter from the FDA with BioMarin's gene therapy, which would have been the first to market, ICER, the US equivalent of an HTA, proceeded with its review. And the top line conclusion was based on their preliminary analysis and a placeholder price. These are US dollars based on the US market, a placeholder price of $2.5 million. It suggested that it would be cost saving compared to the high cost of current factor VIII at dosing levels that would optimize care. So this is encouraging that gene therapy could be deemed cost effective, but certainly it matters how we get there. And just because a drug is deemed cost effective, it does not mean that it is affordable. And so manufacturers will need to look at the pricing and to make sure their price does align with the value that the drug brings forward. The uncertainties in the data and the robust scientific inquiry that we're calling for certainly will be important parts of making drugs affordable. And so we are needing and calling for new methods of technology assessment that capture outcomes and value beyond those traditionally captured. So looking at the societal value and the other cost offsets in the burden of the disease. The good news from the ICER review is that it actually included placeholders for many of these, and all the data was unavailable, including some of the core outcome data that we called for years ago that we asked manufacturers to have ready for submission and was not. We think that there is opportunity for us as a community to do additional robust analysis, data collection, and strengthen the case. So the pricing in the US certainly is higher than many parts of the world. Context will matter. And what I've discussed in the US context that may be applicable to other developed economies, we will require different strategies for the rest of the world. And certainly there are thoughts ongoing about how to achieve that. But in terms of the typical health technology assessment, I think there is a good news story to tell, but much work still to be done.
Thanks, Mark. Before we go to the question and answers from the participants, I'm going to bring everybody in for some quick final thoughts. And again, to you first, Mark, there's been so much talk about the unknowns and the uncertainties. You know, how do you approach that? And how do you deal with all of this uncertainty yourself?
So absolutely, there's absolutely no doubt that there will be uncertainty, and we cannot expect to have everything that we've talked about today answered on the day that a product is commercially available. If we wanted all of these answers, gene therapy would never be available. So there is an assumption that there will be a degree of uncertainty. But when there is a lack of scientific consensus around those uncertainties, you know, we shouldn't rush to commercialization or have an undue urgency to bring something to market. Where there is uncertainty, we need to make sure that there is the foundational research that's undertaken. And a number of companies are to be credited. They are working on a number of these aspects, but we certainly need more. And we don't want to end up where there is uncertainty in the decision making. The uncertainties really highlight the importance for longitudinal data collection. So many of the things such as variability, durability, integration, the potential for other comorbidities, those can only be answered. The small number of patients that are in gene therapy trials, it's just too small to find those rare events. But we want to anticipate, and those things that are known uncertainties, we should have a plan to look for them, and where possible, we should be investigating now and laying the foundation so that if a question arises, we can answer those. The duration to address the uncertainties may well be different for payers. So we have to bear in mind the balance between the level of uncertainty accepted for patients and the level of uncertainty that will be accepted for a payer. If there's too much variability and too much uncertainty and durability, that could be a factor on reimbursement that's, you know, above and beyond the uncertainty that we as patients are willing to accept.
Thanks, Mark. So our paper is calling for more scientific inquiry. Glenn Pierce, what would you say are the key questions scientists need to be exploring?
Well, I would start off with the general statement, David, and go back to politics. We have a term coined here in the United States, unfortunately, over these last 4 years, called alternative facts. And if we move this to science, there's no room for alternative facts. There are facts and there are facts, and that's it. It's only facts. That's science. How we interpret those facts is, of course, a matter of individual choice. The two examples that I want to give are, one, what's written for investors and what's presented scientifically. There's a huge dichotomy between that. I read something for investors and the world looks rosy, and then I look at the data and it doesn't look so rosy. So can we just be honest about that? Investors aren't stupid. We're not stupid. We need one set of facts. The other part of that is that in some of these scientific presentations that are being constructed by the companies, the words that are coming out of the presenter's mouth are not the same as what's on the slide. If I see data that shows me what a level is at 6 months and another level that's lower at 18 months, don't try to tell me that that's a durable response. And that's exactly what's been happening between the ISTH this year and ASH. We need facts that are reliable and scientifically based and honest. And we expect that. And in fact, I would say we demand that. Let's cut some of this nonsense out and just deal with this in the way it is. It's real life. The data aren't perfect, and we need to be able to address the imperfections in the data in order to make progress to the next stage. We're talking about generation 1 gene therapy here. It won't be for everyone, and it is certainly not going to be the end. There will be generation 2, generation 3, generation 4, AAV, and there will be non-AAV alternatives that are emerging from preclinical work. And so let's just all be honest with one another at this point in time. We need that in order to be able to help every individual patient make the decisions that are needed. The questions remain. More questions, as I mentioned before, are there. And the companies, by and large, are doing very little to answer these scientific questions. But if we don't get the scientific questions answered about variability and durability, we are never going to get to the next stage, which is fixing those problems so that we have a reliable method for curing hemophilia. That's the goal here. That's the only goal, a reliable method for curing hemophilia. How are we going to get there? We're not going to get there by rushing through phase 1, 2, 3 clinical studies, not answering any of these questions, and producing products that produce high variability and a lack of durability.
So meanwhile, patients are being recruited for phase 1, 2, and even 3 clinical trials in gene therapy. Brian Wamani, what is the importance of the informed consent process, given all that we're talking about today?
This is the first irreversible therapy. So unlike any therapy that's come before this for hemophilia, this is an irreversible therapy. You get one chance at this. You do not want to have buyer's remorse afterwards. You can, of course, go back to other treatments, but it is irreversible. So I think what you need is clinicians who are fully conversant with every aspect of gene therapy, who can participate with the patients in a well-structured framework of shared decision-making, and it has to be shared decision-making, where they can discuss the knowns, the unknowns, other treatment options, pre and post gene therapy, and they can look at the requirements for follow-up, both during the trial and for a lifetime, look carefully at the risks and benefits, define the expectations, prepare, and the person with hemophilia going into a gene therapy trial should be prepared, obviously, for a good outcome, but also for a poor outcome and for suboptimal outcome. And I think you need high quality education tools, which need to be both comprehensive and comprehensible to people at different education levels. And I think you also need expert centers to give this information, to work on the shared decision making, and to give the gene therapy. And for that reason, in Europe, there has been consensus between the clinicians and the patient organizations on the need for a hub and spoke model. So we do not envisage too many centers in each country being gene therapy dosing or supervising centers. We see a hub and spoke model where the patient will go to an expert center where every aspect of the decision can be discussed in detail before this irreversible decision is made.
Thanks, Brian. So we've discussed safety and efficacy. Radek and Kaczmarek, what mechanism need to be put in place to monitor safety and efficacy, and for how long?
Thank you, David. Mark touched on this in a little bit, and I'll elaborate. There is an urgent need to establish a mechanism to collect long-term data on safety and variability and durability of efficacy. Some data will come from open-label extensions of ongoing phase 3 and future phase 4 clinical trials. However, much of the burden will be on registry studies to collect long-term data on a large cohort of patients. Clinical trial data can only provide so much. They can provide reassurance of short-term safety and efficacy of specific gene therapy products. But as with any new therapy, in the beginning, we have limited data on the longer-term impact of gene therapy. And to that end, we will need longitudinal registries, which will be the most robust means of revealing unexpected or rare events, detecting low incident or delayed safety events, particularly in small treatment cohorts of a rare disease, which Hemophilia is, necessitates that each patient who receives gene therapy be followed over the long term, preferably over their lifetime. The Hemophilia organizations and clinician organizations have laid a lot of groundwork for that, and so did the WFH by formulating a world gene therapy registry that aims to collect a standardized set of core data developed with input from a multi-stakeholder steering committee that Mark mentioned. And the aim of the WFH Gene Therapy Registry project is to provide a robust, scientifically valid data collection avenue available to all healthcare providers treating patients who receive gene therapy. The Gene Therapy Registry will collaborate with individual hemophilia treatment centers and existing gene therapy registries to leverage established data repositories. Thank you.
Thanks, Radek. First question from the participants, and I'll direct this to Glenn Pierce. Is there a best age to receive gene therapy?
Well, not necessarily, except for the fact that children cannot receive AAV. Maybe I should rephrase that. The best age to receive gene therapy is in an infant, before there's any damage at all to the joints, before there's any risk of an intracranial hemorrhage. But we're not at that point. Number one, because we don't start clinical trials in infants until we know that the whatever the material is safe in adults. Number two, because the infant is rapidly growing and their livers are rapidly growing. AAV therapy is not likely to work in infants, and so the most likely outcome of that is that some non-AAV alternative is going to be required for infants, and that doesn't exist yet.
I'll direct this to the entire panel.
David,
what are the expectations? Go ahead, Mark.
No, I just want to say, because I think Glenn answered the clinical question from the patient perspective, but there is an important caveat as we think about the optimal age, as we start to think about payers, because payers may intuitively think you need to do it at the youngest age. And if a gene therapy does not have a long durability, the argument that we have made to payers is that patients should control the decade in which they want to have gene therapy. It may make sense that when they're 18, it may make sense when they have a young family, or when they're starting their career, or when they're older and at risk of falling and have difficult venous access. So we want to resist being pigeonholed into there is one right age, and make sure we preserve patient choice if we don't have long-term durability.
I'll let anybody answer this next one. What are the expectations in terms of annual bleed rate and factor activity level associated with gene therapy in the community?
Well, I think in terms of annual bleed rate, that's a poor measure, because with currently available therapies, extended half-life factors, emicizumab, and so on, you're seeing annual bleed rates heading towards zero. So you're not going to see a big difference in many cases between the ABR on gene therapy and on current therapies. In terms of factor expression level, I think we're seeing a lot of variability there, as we've seen. I think the crucial thing is that the individual has an expectation before they go into the trial, and hopefully you could achieve somewhere. And problem at the moment is there's really no predictability about what factor expression you will get. So that's why we're looking at variability and predictability very closely. It needs to be matched to the individual's expectations where possible.
Other comments on that question? All right, then. Glenn Pierce, you mentioned this briefly, the question of integration. So how high do you think that risk is of, first of all, integration, which does happen, but more importantly, mutagenesis?
Well, the risk of integration is 100%. And as I said, there are millions of integrations that occur within the chromosomes inside of our hepatocytes and actually inside of many other cells in the body. When we administer AAV systemically, it doesn't just go to the liver, it goes to a variety of other cells throughout the body. It only works in the liver because the way we design the gene, it's only activated inside of the liver to make factor VIII or factor IX. So the integrations occur. Most of the integrations that have been identified in animal model systems are going into places in the chromosomes that don't matter. There's very little to no risk at all of any sort of problem coming from popping a gene into this part of the chromosome or that part of the chromosome. But the risk is that in one particular case, an integration could go into a hotspot in a chromosome and induce a malignant transformation or cancer. And so that's a theoretical risk. There's been a lot of work done in mice. Mice are not human, and so I don't want to put too much emphasis on it. But it's enough of a risk that it means we need to be aware of it and we need to be looking for it. Nothing has been seen in any human clinical trial of any AAV therapy for any disease that would demonstrate that is a likely outcome.
Thanks. So based on the durability considerations discussed today, do any of you have an idea of what would be an acceptable durability period which maintains efficacy?
Yeah, but we've surveyed some people with hemophilia, and some of you indeed at some of the meetings, small groups of well-informed people with hemophilia. What I've seen generally that people are saying either at least 10 years or 5 to 10 years, not less than 5 years, and ideally a lifetime. But I think in many cases now we accept that it's not going to be a lifetime duration of expression. I would say a minimum 5 years, but ideally at least 10 years.
Other thoughts on that?
Well, I would say lifetime. I'm looking for a cure for hemophilia. I don't want a cure for 5 years or 10 years. I want a cure for the rest of my life. It's that simple.
All right, we've been talking about hemophilia A and B for the full hour. Are there any advances in gene therapy in other bleeding disorders besides hemophilia A and B? Glenn.
Other disorders. So there's a lot of work ongoing with AAV gene therapy in other disorders, but hemophilia has been at the forefront because in many ways it's been the most attractive genetic disease to pursue for the last 30 years. And there's many reasons for that we don't need to get into now, but that has attracted a lot of scientific investigation. So most of the new discoveries that are being made in terms of variability, durability, that sort of thing are being made in hemophilia. We don't know how generalizable that is for the other disorders, but as more data come out on other disorders, they will learn from us, and we, I hope, will be able to learn from them on best practices.
I think the question was related to other bleeding disorders. Von Willebrand disease,
okay.
the factor deficiencies.
And so we have a fundamental problem in how drugs are regulated in around the world. A specific drug has to go through an involved process, and ultimately it costs $500 million to $1 billion to get a drug to the market. And so that's not feasible for factor VII deficiency or factor 12 deficiency or 11 deficiency. And it's just not going to happen that way for those deficiencies. There aren't enough patients to be able to support multi-$100 million investments. What we need to be able to do is to develop a system where we can swap out the the gene cassettes. So everything else stays the same. We have an AAV vector that stays the same. We take out the factor VIII gene cassette, and keeping everything else the same except for the gene, we pop in the factor VII gene cassette and then use that and have an expedited kind of an approach toward, for instance, treating a factor VII deficient patient. That hasn't been done yet, but there have been some steps in that direction. Unicure has swapped out one amino acid for another amino acid and has been able to, with minimal loss of time, move to a different product for the treatment of hemophilia B. And so that's the sort of an approach that needs to be further developed in order to be able to get to the rare bleeding disorders. With regard to von Willebrand factor, that factor is too large to put into AAV, and it's highly likely that some other gene therapy system is going to be required to deal with, for instance, type 3 von Willebrand disease.
So here's a topical question. We've heard that some of the COVID-19 vaccines in development might use the AAV vector to deliver the vaccine. Is this true? And what are we doing to make sure people know about that, if it is? And again,
Well,
Glenn, I think you're in a good position to answer
okay,
that.
I've tried to answer that question in chat. For the COVID vaccines, we have these mRNA vaccines, messenger RNA vaccines that two companies are developing. They're the ones closest to approval in a variety of geographies and are going to be the first ones out there. No issue there at all with regard to hemophilia, other than the fact that they need to be given by intramuscular injection, and so we need caution there. There's another vaccine that's in development that uses a cold virus, adenovirus, unrelated 100% to AAV, even though the name is associated. AAV means adeno-associated virus, but it has no resemblance at all to adenovirus. So then the vaccine is put inside this cold virus, adenovirus, and there's no problem there. That one's moving along through the clinic as well. And then there is some discussion about using AAV to deliver the vaccine. That's still in very early development. I'm not quite sure where it's at, but it's not close to approval or anything like that. If you think you ever want to get an AAV gene therapy for hemophilia, do not take that vaccine. Stay away from it, because it'll have the same thing. It'll accomplish the same thing that we've already been discussing. You'll get very high immune responses to it, and you'll never be able to take another AAV if you take that one.
And Glenn, I'm sure WFH will be communicating if there ever is an AAV vaccine. We'll make sure people know about it.
Yes.
Good. And one last question for Radek. The last question. Could you clarify where we stand with the gene therapy registries for hemophilia A and B? Have they already been established on a global scale?
Well, I actually think that Glenn is in a better position, more up to date to update the audience on this.
So the registry, the registry is in the process of being established. We have spent 2 years working through the data that will be collected. It's been a multidisciplinary effort. The ISTH has been our full partner in validating the data collection and in identifying all of the categories. We have also worked with a number of other organizations, EHAD, the EHC, the NHF, NHF-MASAC, and ATHEN, which is the American Thrombosis Hemostasis Network that collects all the data on individuals in the United States. And we've actually paralleled with ATHEN to develop the same core data set for use in a gene therapy registry. We're working with our industry partners as well and have their input and buy-in to the questions that will be collected in the registry so that it's going to be comprehensive. Our goal is to collect information on every patient with hemophilia that gets gene therapy. We recognize that that may not be able to happen 100% of the time, but the more patients we have, the more likely it is we're going to find a problem along the way, if a problem exists. And so that's going to be the importance of the registry, to be able to do long-term follow-up. Mark's got a point to make.
So just sort of building on what Glenn said and sort of the other conversations, one of the groups that we've not mentioned at all today are our regulatory partners at the FDA and the EMA. And I think it's important, just for what Glenn was talking about, to say the FDA and the EMA have been active discussants in the development of the registry so that it would hopefully support their needs. There have been at least 3 publications, and you could get those from the WFH on the gene therapy registry and the core outcomes. I think it's also important to note, in the core outcome discussion I had earlier in this session, the FDA and the EMA both participated in the development of those core outcomes. As patient organizations, we have unique and strong relationships with our regulatory partners. And throughout all of this, it's not as if we're just talking with those listening to the webinar today or among ourselves, but we're actively engaged really on a policy front across the board, and have sought collaboration and shared our opinions on many of these subjects directly with regulatory authorities, both product specific as well as generically about what we expect and hope that they will be doing long term.
Thanks, Mark. So I know we didn't get to all the questions, but we'll keep those questions in the archives and find other ways to answer them. So if we perhaps move to our last slide, which is the key message. And so I think this is what the 6 authors of the paper in hemophilia wanted to say. We wanted to send an urgent reminder that while gene therapy is under clinical investigation, we need more transparency from all those pursuing clinical trials, including meaningful research on the unanswered questions around safety, variability, and durability of response. Only in this environment can we have confidence in this technology. So I hope you have enjoyed the webinar and learned something about gene therapy. In closing, I want to thank the World Federation of Hemophilia for hosting this session. And I especially want to thank our 4 panelists for their skill in translating very difficult concepts into something we can all understand. So wherever you are in the world, have a great day, evening, or night. Thank you.